Welcome to another video where we are now going to discuss the concept of immunoglobulin class, as well as the various effector functions that immunoglobulin and especially antibody mediate in their role as antimicrobial effector molecules. If we just sort of think about class in the most simplistic terms, we can see that antibodies sort of have five major flavors that they come in, and some of these you may have heard of before. You can see across the top here that the five classes of immunoglobulin are IgM, IgD, IgG, IgA, and IgE. And you can notice some differences immediately in terms of their structure. Some of these different classes are the sort of traditional version that we think of, just the classic Y shape, whereas classes like IgA and IgM are actually multimers of that shape. So IgA is often found as a dimer of two sort of immunoglobulin molecules, whereas IgM often exists as pentamers or hexamers, and for that reason is actually has a high degree of avidity, as we saw in the last video. But what is really structurally different among these different classes of antibody isn't really whether or not they are a monomer or not. It has to do with the structure of their heavy chain. And there are essentially five different types of immunoglobulin heavy chain that can be incorporated into the immunoglobulin molecule, and the heavy chain is what determines which class the immunoglobulin is. So the heavy chain that corresponds to IgM is designated as mu, and that in fact is where the M in IgM comes from. Mu sort of starts with the letter M, sounds like an M, and you can apply that logic across the board here. IgD has a delta heavy chain, IgG a gamma, IgA alpha, and IgE epsilon. And this is where being able to recognize these Greek symbols can be very helpful for any student of biology. Again, the heavy chains are referred to by their Greek symbols, whereas the class of the antibody uses the sort of the more common letters like M, D, G, A, and E. Another thing that I've pointed out here on this slide is the sort of relative frequency of these different classes as they are found in the serum of an average human being who is otherwise healthy or normal. And you can see that by far the most common antibody that we find in the serum is IgG, which is one reason that many people have heard of it and maybe haven't heard of the others. And we will see reasons later in the course why IgG is the most abundant. And it's a very important immunoglobulin because it mediates many very important antimicrobial effector functions, whereas some of the others are a little bit more esoteric or specialized for various reasons. You can see that moving down from IgG, the next most common isotypes that we typically find are IgM and IgA, which together represent, each maybe 10 to 15% of the overall immunoglobulin that we find in serum. And then from there, we move into even much rarer immunoglobulin types like IgD, and then the rarest, at least in the so-called industrialized West, will be IgE. And this one reason for that is that IgE is typically specialized for parasitic responses, as we will talk about in just a few minutes. But overall, I just sort of want to point out that each of these immunoglobulin classes is specialized for a different set of functions, and therefore they are all represented in the serum of any given human being at different ratios and sort of correspond with how much we need them and in what context. So a couple of points here that I want to make is that here we are looking at antibodies, but remember that immunoglobulins can serve as either antibodies or as BCRs, B-cell receptors. And if we look at each of these classes when they serve as B-cell receptors, we can see that when a naive B-cell starts its life, the class of its B-cell receptors is actually both IgM and IgD. That is to say that the BCRs of a naive B-cell, there's a balance, there's a co-expression of both IgM and IgD BCRs on a B-cell that has never seen its antigen before. Um, However, once that naive B cell sees its antigen and becomes activated, it can do something that we call class switch and start to express BCRs that belong to the other classes like IgG, IgA, or IgE. And typically, it's the type of pathogen, the type of immune environment that the naive B cell is exposed to that is going to determine which class it might switch into later. For example, viral infections tend to skew naive B cells into becoming into class switching into IgG because IgG is more helpful for viral infections. Where again, parasitic responses tend to influence class switch towards IgE because you need more IgE during a parasitic infection. A couple of notes here, though, is that once a naive B cell class switches away from IgM and IgD, it can never go backwards. Once it starts expressing a class switched isotype like IgG, A, or E, that is typically a permanent change. There is no co-expression. A naive B cell that expresses IgG only expresses IgG and does not express any other isotype. The same is true for IgA and for IgE. Those sort of rules of thumb are a little bit of an oversimplification, but I think they're enough as we just initially get used to the idea of the various immunoglobulin classes and especially how they serve as BCRs in this context. The sort of weird thing that you may notice here is that naive B cells uniquely co-express both IgM and IgD. And I noted before that the expression of IgD antibody was actually quite rare in the serum. That's because IgD is a little bit of a mystery. We don't really fully understand what the purpose of IgD is. IgM is by far much better studied and much better understood in terms of its function, both as a BCR and especially as a secreted antibody. But nevertheless, IgD still occurs in nature. It may, in fact, be vestigial, meaning sort of left over in evolution and its function sort of lost to us in time, or we may discover previously unappreciated roles for IgD that just really haven't come through in the science yet. But just a note about it is that there's really not much to say about it beyond the fact that it is co-expressed with IgM to serve as a BCR on naive B cells. If we think about all of these different classes, again, switching our attention back to the antibody versions of these molecules, I said that class is all about the heavy chain. What's especially important about the heavy chain is not only the variable region, which interacts with the antigen, but remember that the Fc domain of antibody is made up completely of heavy chain. And the Fc domain is what is going to confer many of the effector functions of antibody molecules. And one way that the Fc domain, which remember is the kind of stalk of the Y, and you can see here that this sort of stalk looks a little bit different for each of the classes. Some of them, for example, are bigger, they have a higher molecular weight than others. But universally, these Fc domains, something they can do, something they share in common, is their ability to bind to cell surface receptors known as Fc receptors. And these Fc receptors are typically on the surface of immune cells. And when these Fc receptors become activated, they transduce some signal into that immune cell, which mediates some type of effector function. And you can see that there are Fc receptors that are specialized or uniquely complementary to the Fc of the different immunoglobulin classes. So IgM, its heavy chain is going to bind to an Fc mu receptor, again, mu corresponding to M, whereas the Fc of IgG is going to bind to an Fc gamma receptor, IgA to an Fc alpha receptor, and so on, IgE to an Fc epsilon receptor. And there are actually subtypes of these receptors. Again, I'm oversimplifying just a little bit. There are many different Fc gamma receptors, for example. But I want to highlight here just there is a degree of specificity in terms of there are unique sets of receptors that recognize each class of immunoglobulin. Notice here that there is no Fc delta receptor. So as I said before, even though we make a little bit of secreted IgD antibody, there is no known Fc delta receptor that recognizes the heavy chain of an IgD antibody. So again, a little bit of a mystery. The effector functions that we do know about are mediated exclusively by these four classes of antibody, which you see here. So in the rest of this video, I just want to talk about a few example effector mechanisms that are mediated by antibody, and the first couple of them are going to be Fc receptor dependent. This is by no means exhaustive. There are many Fc receptors. They have very complicated signaling. They are expressed on a variety of different cell types, but I'm going to give you just a couple of examples to sort of highlight the idea of the types of things that Fc receptors can do to shape an immune response, and by extension, the way that antibody then serve as effector molecules, because they sort of unlock these effector responses through their binding to Fc receptors. The first is the induction of phagocytosis. So in a previous video, we saw that Fc receptors were actually examples of receptors that induce phagocytosis on the surface of phagosomes. So we're coming back to that point now with a little bit of additional context. We can see that particular classes of antibody, especially IgG and IgA, are actually good opsonins. When they coat the surface of a pathogen, that is a clear signal that we want to phagocytose that pathogen. The way that is accomplished is through Fc receptors that are expressed on the surface of phagocytes. In this example, you can see that IgG is bound to the surface of this bacterium, and that when it binds to Fc gamma receptors on the surface of the phagocyte, let's say this is a macrophage, that's going to be a strong signal to induce internalization of a phagosome, to eat that bacterium. One thing that I want to note here is that typically Fc receptors require something called cross-linking to become activated. So we really rely on antigens that recur on the surface of pathogens, meaning that these are very common antigens that happen over and over again or expressed over and over again, so that multiple antibodies can bind to the surface of the pathogen. And when multiple antibodies bind to the surface of the pathogen, if their Fc receptors then bind to, if their Fc regions then bind to Fc receptors on the surface of the macrophage, that tends to sort of force those Fc receptors to come into close physical proximity with each other in the membrane. And that physical proximity facilitates downstream signaling events, which I have not shown here just for simplicity's sake. But this cross-linking activates particular kinases and downstream second messengers and transcription factors and so on that unlock various effector functions like, for example, phagocytosis. So key to know that Fc receptors typically function through cross-linking, and on the example that we see here on the slide, one of the results of the cross-linking of Fc gamma receptors is the induction of phagocytosis. But we could say the same for Fc alpha receptors bound to IgA. I want to make the point that the cell types that express the particular Fc gamma and Fc alpha receptors that tend to induce phagocytosis are those cells that are famously phagocytes, like macrophages, monocytes, dendritic cells, and neutrophils. You may recall that macrophages, monocytes, and dendritic cells collectively as a family were known as mononuclear phagocytes. One of the reasons they are such good phagocytes is that they are rich in the expression of phagocytosis-inducing Fc receptors, and then that's also true for neutrophils. So phagocytosis is one Fc receptor dependent effector mechanism induced by antibody. Another major Fc dependent effector mechanism is actually the degranulation of various granulocytes of the innate immune system. So you may recall that cells like eosinophils, basophils, and mast cells are particularly important anti-parasitic effector cells. And I also said that IgE is especially important for fighting parasitic infections, and this is where those two ideas sort of link up. These particular granulocytes express on their surface very high affinity versions of Fc epsilon receptors. And in fact, this functions a little bit differently than what we just saw with IgG. These Fc epsilon receptors are in fact so high affinity that they actually bind to the Fc of IgE, whether or not it is actually bound to any antigen. And so in fact, the IgE antibody is usually just sort of sticking off the surface of the granulocyte because it's being held, it's being tethered by its very high affinity interaction with the Fc epsilon receptor. So in this case, this basophil is sort of just going around the body, sort of holding out the IgG, sort of using it almost kind of like a tool to sample or surveil the environment around it for any particular antigens that might bind to those IgE molecules. And when, for example, it comes into contact with some kind of parasite that has an antigen that the IgE can bind to and recognize, that will also induce cross-linking of the Fc epsilon receptors. And the cross-linking of Fc epsilon receptors is a strong degranulation signal. And eosinophils, basophils, and mast cells, their granules contain very effective anti-parasitic molecules, various protease enzymes, as well as things in the case of basophils and mast cells like histamine, which mediate kind of physiological responses which help us clear parasitic infection as well. So again, just another example of an Fc receptor dependent effector mechanism mediated by antibody. But in addition to activating Fc receptors, antibody, even without those interactions, has some additional effector mechanisms that it can mediate. And so I'd also like to talk about just a couple of examples of those. One that we can sort of now come or bring full circle to a previous video is the initiation of a very important innate immune response called the classical complement pathway. So you may recall that the classical complement pathway, the one thing that made it unique was that it was initiated by antibody rather than by a lectin or spontaneously starting. So the two classes of antibody that can initiate the classical complement cascade are IgM and IgG, as you can see here. And this does not require Fc mu receptors or Fc gamma receptors. Instead, what happens is that when these two classes of antibody bind to the surface of a bacterium or some other pathogen, they serve as a substrate for C1q to bind. And when C1q binds to antibody, that sort of links it to the pathogen through that binding interaction, but that also induces a conformational change, which activates the protease domains of C1, which were C1r and C1s. That allows them to cleave C4 and C2, as we've seen before, and that initiates the classical complement cascade, which can unlock the effector mechanisms of that pathway. Something to note here is that while both IgM and IgG are capable of initiating the classical complement cascade, IgM is actually a bit better at it, and that is because it forms these really large pentameric or hexameric structures. And when the arms of this structure actually bind to the surface of the pathogen, it can form something that we call a staple. It sort of serves as a signaling platform where it's sort of held on at multiple locations. And this is a pretty big overall molecule that then C1q can dock to. And in fact, a single IgM molecule, because it is so big, can effectively serve as its own platform for the binding of C1q. In contrast, IgG can bind to C1q, but you need multiple IgGs bound close to one another to serve as an effective scaffold for C1q binding. So while both IgM and IgG initiate the classical complement pathway, IgM is a bit better at it and therefore is a bit more specialized for complement activation. Okay, one additional Fc receptor independent effector mechanism that I'd like to discuss is something unique about the class of antibody known as IgA. We saw again in a previous lecture that IgA was particularly enriched at mucosal surfaces. It helps us fight the pathogenic bacteria, especially that are attempting to colonize our barrier tissues, especially those that are covered in mucus. The reason we find IgA there in such abundance is because there are some specialized mechanisms that help us secrete IgA into the mucus, where it can then serve as an antimicrobial effector at that site. So if we take, for example, here, this is a cross-section of the gut. So here you can see the lumen or the open space kind of inside the tube of the gastrointestinal tract. You see that the walls of the gut are covered in mucus and structurally are made up of gut epithelial cells. On the basolateral surface of those epithelia, there is a receptor known as the polymeric immunoglobulin receptor, or PIGR. Polymeric immunoglobulin receptor binds to polymers of IgG, which is what that name suggests, and the polymeric IgG in question is going to be IgA, which exists as a dimer. IgA is being made by cells in a site called the lamina propria, kind of, the other side of the walls of the gut in this example. This is actually an example of a plasma cell, which is making IgA. But this IgA dimer One of the reasons it exists as a dimer is because this covalent link between the two monomers that causes it to become a dimer actually stabilizes this molecule. And it's going to need help remaining stable because the mucus is actually a pretty harsh environment. There's a lot of proteases and other types of enzymes there, which we use actually to sort of control the bacterial growth that happens in that location. But the immunoglobulins themselves, the IgA molecules themselves, are also susceptible or can be susceptible to that harsh environment. And so this dimerization reaction gives them a bit more stability. But in addition to being a dimer, when IgA binds to the polymeric immunoglobulin receptor, that receptor induces endocytosis in the epithelial cell, and that endocytosis actually goes all the way across the cell in a process called transcytosis. And so the epithelial cell actually takes the IgA from the lamina propria and spits it out on the apical surface of the epithelium into the mucus through the process of transcytosis. And following that transcytosis event, a big portion of that receptor actually remains attached to the dimeric IgA and is now something that we refer to as secretory component, or SC. So IgA dimers bound to secretory component have additional sort of protection, additional stability, which is going to help them maintain increase their half-life in the harsh environment of the mucus layer at the mucosal surface. And we rely on these IgA complexes to help us do things like regulate the microbiome, because they tend to target pathogenic bacteria that may be colonizing or trying to colonize the surface. They have other regulatory functions in this site as well. And this does not only happen at the gut, but happens at other mucosal surfaces as well. And interestingly enough, a similar process also happens in the epithelial cells that make up the lacrimal tissues in breastfeeding people. And so IgA can actually be transmitted or transcytosed actually into the breast milk as well. And so IgA is one form of passive immunity that can be passed on to a nursing infant. And a very similar sort of molecular processes happen at that location to facilitate that as well. So another unique feature of IgA. Okay, let's summarize this discussion. We saw that the Fc regions of antibody bind to immune cells, as well as some other cell types, but primarily immune cells, in order to induce various effector functions. These Fc regions bind to specialized receptors called Fc receptors, and we saw some examples of the types of things that Fc receptors induce, like phagocytosis, as well as degranulation. We saw that antibody immunoglobulin generally is divided into five classes, which are determined by the particular isotype of the heavy chain. We saw that there were IgA, D, E, G, and M. Finally, we saw that the different antibody classes all have their unique Fc receptors that correspond to them. So, for example, IgG binds to Fc gamma receptors, whereas IgE binds to an Fc epsilon receptor. And importantly, these different Fc receptors are going to be specialized for mediating different signaling outcomes. And it's going to help us sort of specialize or tune an immune response to the type of pathogen or the type of situation that we're dealing with. And a point that I did not make is that different cells express different combinations of Fc gamma receptors. So as we saw, things like macrophages are going to express Fc gamma receptors, which induce phagocytosis, whereas a basophil is going to have primarily Fc epsilon receptors to induce degranulation. And again, there are many other examples of unique combinations of FCRs on the surface of various immune cells that sort of correspond to the types of effector mechanisms that those immune cells are important for mediating. Okay, so that covers it for immunoglobulin class. In our next video, we will switch our attention away from immunoglobulin and B cells back over to T cells. See you in that video.